Okay, shall we start our lecture? Okay, sisters and brothers, hope you all are fine. And this lecture, we cover the types of adrenal hormones and then their structures. And I go detail about the biosynthesis of mineralocorticoids, glucocorticoids. So mineralocorticoids and glucocorticoids are called corticosteroids. And later, and I will also try to include the adrenal sex hormones biosynthesis and genital sex hormone biosynthesis. And later, and provide their biomedical importance. And then the lecture will end with the adrenal medulla hormones biosynthesis. Okay, my part is biosynthesis. So as you all are aware, so anatomy of histology of adrenal glands. So anatomy, so this situated above the both kidney. There we have two suprarenal adrenal glands. It's covered with the capsule. And then later under the capsule, there are the cortex, and innermost part is the medulla. And even in the context, when we visualize it, and then you can see that there are three main layers of the cells present in the cortex. You can see that zona, outermost layer is zona glomerulosa. Intermediate one, in the next to the zona glomerulosa is fasciculata, and then next one is reticularis. Later you can find the medullary parts. So that's why the mineralocorticoids, aldosterone or deoxycorticosterone, so they are synthesized in the outermost layer, zona glomerulosa. So function is very important. You have studied the function detail of mineralocorticoids in year one, right? So it's regulate the sodium and water retention and potassium loss as well. So the middle layer, zona fasciculata, allow the glucocorticoid synthesis. And of course, glucocorticoids is anti-inflammatory agents that involve in the gluconeogenesis and protein synthesis as well. And then zona reticularis and middle layer, and they also provide the DHEA, dihydroepiandrosterone, and then also androgen as well. So you can see that these three layers, the three layers of cortex provided the various types of important hormone synthesis. So starting material is actually cholesterol. So cholesterol is the mother compound that provide the various types of steroid hormones biosynthesis. So you can see that here. So the aldosterone synthesis is relying on the stimuli and trophic factor. So you have already known that renin-angiotensin aldosterone systems is the main regulator that support the aldosterone synthesis. Of course, ACTH, adrenal cortical hormones, its role is very important in the synthesis of adrenal cortical hormones, not only aldosterone, but also to the cortisol. Okay, and then cortisol synthesis also rely not only on the ACTH release, also corticotrophin that is synthesized in the hypothalamus. So reticularis provide the DHEA and androgen. So they are hormone synthesis initiated by the influence of ACTH, LH, and FSH. Okay. So medulla hormone. So medulla hormone major stimuli are the stress, fight and fright reaction, as you have already know. So adrenaline, noradrenaline, and then dopamines are the catecholamine that are synthesized in the adrenal medulla, but main hormones synthesized in the adrenal medulla is adrenaline and noradrenaline. So adrenaline increase the heart rate, blood pressure in terms of physiological functions, norepinephrine, it's constrict the arterioles, and then by doing so increase blood pressure and all that. Okay, my dear sisters and brothers, since I'm sharing you the biochemistry, I cannot avoid the structure, biochemical structures. And then, but the detail structure activity relationship will be delivered by the Dr. Zalika in her medicinal chemistry part. So let me introduce the structures or adrenal steroids. Of course, the mother compound is cholesterol. You can see that there are the 4 ring, ABCD ring attached with the long chain carbon. So there are altogether 27 carbon give rise the cholesterol structure. You have already known the cholesterol synthesis since in your first year, right? So here number 3 carbon attached with the hydroxy group here. Okay, so this is the mother compound. Later, the side chain will be clipped mean removed, and will form the aldosterone that contain 21 carbons, cortisol, corticosteroids. And later, the lyase enzyme will degrade the carbon number 17 and 20, 17, 20 lyase, and give rise to form the androgens, estrogen later. So you can see the carbon change and androgen is the longer than the estrogen. Okay. Okay, associating. So mode of action will be explained here. Okay, the structure detail again. So you can see that basic steroid nucleus, and later you will form glucocorticoid series here, and then androgen series here, 19. Okay, so again, you can see that the structure is a corticosteroid structure. Okay, corticosterone. Okay, aldosterone, structural aldosterone. You can see that testosterone, progesterone, and then the beta estradiol or estrogen. Okay, so the synthesis. So as I mentioned earlier, main stimuli for these cortical hormone synthesis are adrenocorticotrophic hormone. So adrenal ACTH secretion in the anterior part of the pituitary is also guided by the corticotrophin, which is synthesized in the hypothalamus, right? So ACTH, once ACTH is secreted from the anterior part of the pituitary, so they are transported to the adrenal cortical tissue. In a cortex cell membrane, so the ACTH bind to its receptor, and then ACTH binding, please refer to your lecture number 1, my lecture. So already deliver, activate the G protein. G protein activation also activate the adenylyl cyclase, and then as a result, cyclic AMP is released from the ATP hydrolysis. So cyclic AMP role, cyclic AMP carry the message of ACTH. So what's the action is the activation of protein kinase A. So protein kinase A activate the esterase enzyme. So since before the hormonal cascade happened, the lipoprotein is carried and uptaken by the adrenal cortical tissues. Okay, receptor mediated endocytosis reaction. Okay, so lipoproteins enter, and once lipoprotein enter into the cells, so lipoprotein are degraded by the lysosome. So cholesterol part, especially cholesterol ester part. So cholesterol ester is degraded by the esterase enzyme. So esterase enzyme activity is, activated by the protein kinase A. You know that. So protein kinase A activation you have already seen here. So protein kinase A will activate the esterase. Esterase degrade the cholesterol ester, and finally you will receive the cholesterol. So this reaction occur in the cytoplasm. Later, cholesterol are transported into the mitochondria membrane. Mitochondria. transport is we require the star protein. What is a star protein? Steroidogenic acute regulatory protein. Okay, this protein, the process is mediated by the steroidogenic acute regulatory protein, and later you can see that it is transported inside, and later it will continue and complete the reaction until the three different types of hormones are synthesized, completely synthesized. Okay. Okay, there are various types of enzymes available inside the mitochondrial membrane. For example, side chain cleavage enzyme that degrade the long carbon change from the cholesterol, make the pregnenolone. So pregnenolone again, isomerization occur. Pregnenolone will change to the progesterone here. The progesterone will go to the three different pathway, and finally we synthesize the mineralocorticoids, glucocorticoids, and androgens. Okay, see how the detail. So there are three main pathway. Pathway A provide the aldosterone synthesis, and pathway B provide the cortisol synthesis. Pathway C, let's say, provide the androstenediol. So you can see that, my dear sisters and brother, cholesterol is starting material. First of all, cholesterol long chain must be removed and remain only nucleus. So pregnenolone, again, oxidation and isomerization reaction occur and provide the progesterone. So by the action of 3-beta dihydrogenase enzymes groups, so progesterone is formed. And then 21-hydroxylase make the attachment of the hydroxy group in the number 21 carbon of the steroid nucleus and make it alpha deoxy 11-deoxycorticosterol. Next one, 11-hydroxylase attachment of hydroxy group at 11 carbon 11 position, and hydroxylation reaction occur, corticosterone is formed. Corticosterone is the intermediate product of aldosterone. The next step is hydroxylation reaction and make aldosterone. You can see that there are a lot of hydroxylation reaction occur in order to make sure the to complete the synthesis of hormone. Okay, when you check the cortisol production, pregnenolone, first of all, 7-alpha hydroxylase reaction, hydroxylation reaction occur and provide the 11-hydroxypregnenolone, and then 3-beta hydroxydehydrogenase enzyme will form the 11-hydroxyprogesterone, 21-hydroxylase add on it, and form 11-deoxycortisol. The same enzyme 11-hydroxylase catalyze this reaction, finally cortisol is formed. Okay, so since they contain 21 carbon in their structure, but in order to synthesize the shorter carbon chain, androstenediol, so 17 and 20 lyase mean degrade the carbon and make it DHEA that contain 19 carbon, and the same 3-beta dehydrogenase add on it and form androstenediol. Androstenediol or androgen, I mean, that can be derived either from DHEA and 11-hydroxy progesterone as well. Okay, so these are the process reaction. So this is pathway 1, classical pathway 1A, and pathway B and pathway C. So in the gonads and sex glands, of course, androstenediol is catalyzed by the aromatase enzyme and form the estrone. Estrone is actually E1. In terms of structural difference, so estrogen can be classified into the E1, E2, and E3. So E1 is derivatives of androstenediol. So how about the E2? E2 can be either derived from the E1 or testosterone. So the male sex glands and then the testes provide the testosterone. The tissue, in the peripheral tissue especially, this is catalyzed by the aromatase and can form the estradiol. So that's why even in the male, they have estrogen E1 and E2, okay? Because the peripheral tissues provide the aromatase enzyme and can form the estradiol. Okay, my dear sisters and brothers. So shall we go the mineralocorticoids detail? So first of all, before initiate the biosynthesis, cholesterol must be important, right? What you call that, present inside the tissues, zona glomerulosa, fasciculata, reticulata, so on and so forth. So as I mentioned earlier, so ACTH stimulates the enzyme esterase. Okay, so esterase degrade the cholesterol ester from the lipoprotein. So as cholesterol and ester group is they detach each other. And then so cholesterol are transported into the mitochondria by the steroidogenic acute regulatory protein mediated mechanisms and shortening or change. So shortening or change is done by the specific enzyme. We call it cytochrome P45017 cleavage enzyme. So this enzyme activity require NADPH and oxygen. Okay, NADPH and oxygen are required for the reaction. So NADPH is very important. You have already known that, right? HMS pathway, hexomonophosphate shunt. In each and every cell, they have enough NADPH. So NADPH is used for the reductive biosynthesis, including the steroid hormone biosynthesis. So, okay, in the endoplasmic reticulum or mitochondria, pregnenolone is formed after shortening of the cholesterol change. So next reaction, as I showed you earlier, oxidation and isomerization reaction. So this reaction is catalyzed by the 3-beta hydroxysteroid dehydrogenase plus 5,4 delta 5,4 isomerase enzyme. It's an enzyme complex. They work together to make sure oxidation and isomerization is done so that the pregnenolone can be converted to the progesterone. So next hydroxylation reaction occur at carbon 21 carbon. So you can see that 21 hydroxylase enzyme form 11 deoxycorticosterone. So please check the detailed reaction in the picture I shown before. Hydroxylation reaction, next step. At 11 carbon by the 11 beta hydroxylase enzyme, so produce the corticosterone. Okay, so corticosterone is actually a weak mineralocorticoid reaction, but it also has glucocorticoid activity. That means it can lower it has anti-inflammatory actions. It has some gluconeogenic action involved in gluconeogenesis. Okay, so and then the last step, the hydroxylation reaction at hydroxylation reaction, and then you can add these 18, carbon number 18, and finally you got the mineralocorticoid. So the main stimuli, as I mentioned earlier, renin-angiotensin-aldosterone system is very important. So, okay, so actually, hello. Hello. Actually, when your sodium potassium ratio in the plasma decrease by the hormone, hormone angiotensin 2, actually this is the octapeptide. So it's produced from the angiotensin 1 by the enzyme angiotensin converting enzyme, right? The enzyme found predominantly in the lungs, also distributed widely in the body. So angiotensin secreted by the liver And then also cleavage is accomplished by the renin, right? So angiotensin 2 bind to the receptors, and then it's a little bit contrast with the ACTH. Its effect is mediated through the IP3 pathway. Okay, IP3 pathway. So aldosterone primarily affect on the kidney tubules, where stimulate the sodium and water uptake and potassium excretion. So angiotensin competitive inhibitors or angiotensin converting enzymes are used to treat the renin-dependent hypertension. So nowadays, this is the first line drug in treatment of hypertension. Okay, so you can see that normal plasma value of aldosterone is very low. It's about 100 to 500 picomole per liter. So shall we go to the glucocorticoids? Okay, glucocorticoids also similar pathway you have already seen. So cholesterol is a major compound. First of all, pregnenolone has been changed, and later progesterone is formed. Isn't it? And 21 hydroxylase add on it, and then release the 11 deoxycortisol. So 11 beta hydroxylase catalyze on it. Finally, the most potent glucocorticoids named cortisol is synthesized. Okay. So as I mentioned earlier, corticosterone is also formed from the 11-deoxycorticosterone by the 11-beta hydroxylase. Okay, so main stimuli, as I mentioned earlier, do not forget about the ACTH is a main stimuli. Okay, so cortisol level is follow the diurnal rhythm, isn't it? So that's why you can find the normal cortisol level we have to measure in the fasting time at 9 a.m. So the plasma concentration of cortisol is about 140 to 690 nanomole per liter. But at the midnight, you can find the least value, plasma value can be detected. It's about less than 100 nanomole per liter, because the cortisol is used up throughout the day to function the physiological function. So all the steroid hormone transport require the carrier protein. So only 10% can be found in free form, but mostly found with the either globulin or albumin. So majority bind to the globulin, and globulin bind it, and then it's transported to its target tissues. So half-life is about 70 to 90 minutes. So innervated mainly in the liver. And all the steroid metabolism occur in the liver. First of all, there is a reduction reaction occur, hydroxylation reaction occur, and then some of the steroids, the conjugation reaction, they undergo the conjugation reaction. So that's why approximately 20 to 30% are excreted in the bile acid, while the rest are through the kidney. Okay, 1% unchanged or free form found in the urine. Okay, my dear sisters and brother. So now I would like to talk about the androgen biosynthesis. Okay, you can see that androstenedione, DHEA is formed, androstenedione and testosterone form, right? So in terms of the reversible. So androstenedione provide the estrone formation by the enzyme aromatase. Similar things happen to testosterone. Testosterone provide the E2 by the enzyme aromatase. So factor that increase the aromatase activities are age. So increasing the age, aromatase activities decline. Obesity also decline, and insulin enhance the, in terms of age, meaning to say that it's a certain age. For example, from the puberty and reproductive age, the age increase and aromatase enzyme activity is increased. Okay, obesity also it increase. Insulin, since it's an anabolic hormone. So insulin also increase the aromatase enzyme modifications. Gonadotrophin and some alcohol action increase the aromatase activities. So in that case, aromatase action, the higher the action of aromatase, the more E1 and E2s are formed. Beware in your mind. This is a reason why that particular male has a gynecomastia problem. Okay, this is aromatase activity. He is C testosterone. So in a estradiol E2, you can see the hydroxy group attached here, okay, and then here. So this is the only one what aromatase changed the testosterone to E2. So structural difference, you can see that methyl group has been removed, and then the what you call the keto group here is attached with the hydroxy group here, changed to the hydroxy group. Okay, so biosynthesis or adrenal sex hormone synthesis, as I mentioned earlier, starting compound is cholesterol. Side chain cleavage enzyme will remove the long chain carbon from the cholesterol, make it pregnenolone. Pregnenolone later by the 17 alpha hydroxylase enzyme. This enzyme are found in the smooth surface endoplasmic reticulum, make it the 17 alpha hydroxypregnenolone, and lyase remove the carbon chain of 17 alpha hydroxypregnenolone to the dihydroepiandrosterone is called DHEA shortly. So 50%, so the interesting thing is that 50% of DHEA, if we do not have a higher demand to form the sex hormones, 50% of DHEA are rapidly modified by addition of sulfate in the adrenals, glands, and stored temporarily, or maybe they are transported in the liver and stored. Removal of the sulfate from DHEA result in the reactivation again. Okay, so if demand of sex hormone biosynthesis is high, DHEA will convert back sulfated form, that is continue the reaction. Okay, so DHEA, 3-beta hydroxysteroid dehydrogenase and delta 5,4 isomerase action, it formed to androstenediol. So reduction again at the 17 carbon resulting in the formation of testosterone. Okay, so aromatase is also like other enzymes involved in the steroid biosynthesis. They are also cytochrome, but species is different, cytochrome 19A1. Okay, the main stimuli for testosterone synthesis is LH and FSH for estradiol. Okay, actually the reduction of androstenedione and then aromatase activities, these are found in the sex glands. Okay, let's talk about the adrenal steroids functions of biomedical importance. So they are very important for glucose homeostasis, especially in the fasting. Corticosteroids, I mean the cortisols, increase the gluconeogenesis pathway. And it's also involved, aldosterone involved in sodium retention, blood glucose, blood pressure regulation. So, and then the corticosteroid involved in host defense, stress response, and general protein metabolism. In the absence of the gland function, it's a life-threatening condition. You may refer to the Addison disease. So the next lecture will be delivered by the Dr. Sandra. I think she will deliver it about the Addison's disease detail. Okay, so glucocorticoid is very essential vital hormones for the adaptation of severe stress. Mineralocorticoids, of course, very important for water and electrolyte balance. Estrogens are not synthesized significantly in the normal adrenal glands, but in the certain cancer may be produced. So bear in your mind, sometimes the plasma estrogen level is high and you know, the feminization symptoms occur if certain cancer, if the person has a certain cancer, I mean, there's some lung cancers also produces the, you know, estrogens wrong place, from the wrong place. So it may also, you can find it, okay? And androgens of the adrenal origins are important precursors of estrogen in postmenopausal women. You know why? The postmenopausal women, estrogen and progesterone production decline, gradually lost. So in that case, the postmenopausal ladies, the adrenal cortex are preserved for the synthesis of androgen. Of course, androgen, when they are transported to the peripheral tissue, aromatase convert the androgen to the estrogens. Okay, so aromatase, okay, as a result, you know, that can be used for the clinical applications. You can see that. So the postmenopausal women with the breast cancer or the problem with the male with the gynecomastia problem. So the role of aromatase inhibitor plays important, you know, because you know why? They can inhibit the aromatase. By inhibiting the aromatase enzyme, there will be no production of estrogen. So estrogen responsive breast cancer are very useful to treat the estrogen responsive breast cancer, similar way the obese men with the gynecomastia problem. So beware in your mind. And prolonged increase of plasma glucocorticoids is caused by the administration of excessive amount of exogenous hormone. For example, let's say chronic immunodeficiency cases and certain autoimmune diseases, and then chronic asthma cases, they rely on the exogenous corticosteroids, right? Intravenous or either prednisolone. So in that case, the plasma glucocorticoids level is elevated. At the same time, ACTH production may be inhibited, okay? And cortical hormone hypersecretion can be found in the adrenal tumors and also the Cushing syndrome as well. So whereby we increase the protein catabolism, hyperglycemia and hypertension may be the result from the Cushing syndrome, as you have already know. And excessive aldosterone secretion is called in the Core syndrome due to the adrenal tumor. They will present with the hypokalemia and hypertension as well. So I think this pathology will be explained in detail by the Dr. Sanda. Okay, so what I want to highlight is that the enzymes plays important role in biosynthesis of mineralocorticoids, glucocorticoids, and androgens. So there are mutation happen in the enzymes. Particular enzyme may lead to the congenital adrenal hyperplasia. Okay, would like to show you these slides. A little bit difficult, but it's once I explain, so you would appreciate it. Okay, here, 3-beta-hydroxysteroid dehydrogenase can be deficient if the enzyme synthesis impaired by the mutations in their corresponding gene. What happened is that, you know, no glucocorticoids, mineralocorticoids, and androgen synthesis because it's blocked since at the beginning of the early step of the biosynthesis. So the salt excretion will be released in the urine and female-like genitalia. This is actually very rare case. It's happened in the autosomal recessive disorders. Okay, if 17 alpha hydroxylase enzyme, cytochrome B17 enzymes are deficient genetically, so no sex hormone and then no cortisols are formed, but you can see that this way bypass, right? So the progesterone will go to the classical pathway A and increase production of mineralocorticoids. So what happened is that sodium fluid retention occur as a result. A person will present with the increased blood pressure. And also they will have a female-like genitalia because androgen is not formed. So the most important and common and common what you call that congenital problem with the congenital adrenal hyperplasia is due to the absence of 21 alpha hydroxylase enzyme deficiency here, 21 here. Okay, what happened? 21 hydroxylase here, partial and virtually complete deficiency, no mineralocorticoids. Glucocorticoids are usually absent, 21 hydroxylase. Okay, 21 hydroxylase. Okay, and then overproduction of androgen. See, androgen production is totally what you call that not associated with the 21 alpha hydroxylase, right? What happened? So overproduction of androgen will leading to the masculinization of external genitalia in female and early virilization in male. So these are the symptoms. So the what you call that another types of deficiency are here, the 11 beta hydroxylase enzyme deficiency. So decrease the amount of cortisol, and then the corticosterone, increased production of deoxycorticosterone causes the fluid retention. As I mentioned earlier, right? Deoxycorticosterone is a weak mineralocorticoid. So you can see that the genetic errors, because the biochemistry, medical biochemistry is supposed to highlight about the genetic errors, isn't it? So that's why congenital adrenal hyperplasia can be classified in the what you call that deficiency of enzymes that involve in the biosynthesis of steroids hormones. Okay, so you may find that the precocious puberties, virilization, mild hirsutism, gynecomastia in the male, you know. Okay, and the female pseudohermaphroditism, so usually related with the congenital adrenal hyperplasia, 5-alpha reductase enzyme deficiency. So they will, in terms of clinical features, so female will complain about amenorrhea, big clitoris like penis, true they have, though they have a uterus. So this by what to call that, they have two genital features in their body. Okay, so the Addison disease is not due to the congenital adrenal hyperplasia. It is the other pathological condition due to the insufficiency of cortisol and aldosterone secretion because of autoimmune diseases. So these are the clinical symptoms will be explained detail in pathology. Okay, so now time to talk about the sex hormones biosynthesis. So the androstenedione and DHEA are formed. They are transported or in the what you call the sex glands, or sex glands itself can form the DHEA and then the androstenedione by the action from the mother compound, what you call that cholesterol. Okay, okay, basic. Okay, so once you realize about the sex hormone biosynthesis in the sex glands, you would appreciate about the basic of basic approach for the contraception. So I think contraception lecture will be explained and taught by the Dr. Raza in my next lecture. So gonadal hormones are anabolic hormone. They are required for the maintenance of metabolism in skin, bone, muscles, and reproductive function as well. So testes, as you have already known, produces the spermatozoa from the Sertoli cell, testosterone from the Leydig cells. Ovary also produces the ova, and sex hormone, estrogen and progesterone, right? They regulate the menstrual cycle and also very important for the reproduction. So as you have already studied the negative and positive feedback regulation, so role of pituitary, hypothalamo pituitary, gonadal axis play important role in the production of these hormones. Okay, so when you check it, you can see that ovary follicle Functional units of the ovum is follicles, and then produces estrogen under the influence of follicle stimulating hormone. The corpus luteum produces the progesterone under the action of luteinizing hormone, and the male stroma testis also produces the androgens by the action of luteinizing hormone and insulin as well. So that's why please refer there. In diabetes mellitus, insulin deficiency or totally lack of insulin occur in the advanced diabetes mellitus cases. Usually men complain about the decreased libido and infertility, impotency. So men with the diabetes mellitus usually complain about that because of insulin deficiency. Okay, so these are the rules, physiological rule of testosterone. You know that. Responsible for the sex drive, produces feeling, aids memory. There are so many functions. You can read that. So the female's function, apart from the reproductive systems, and then the estrogen provide the strengthening the bone, so to prevent the osteoporosis, lower the LDL cholesterol. So that's why ladies has less risks of atherosclerosis and myocardial infarcts and hypertension. So raise HDL and reduce the menopausal symptoms, blah, 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 blah. That is a bad effect of estrogen. Okay. Okay, my dear sisters and brother. So this slide is again showing the structure of E1, E2, and E3. E1, okay, the hydroxy group attached. E1, E1. And the E2, there is a 2 hydroxy group. E2. E3 estriomine contain 3 hydroxy group. Because of attachment of the hydroxy group, estrogen can be classified into the E1, which contain only 1 hydroxy group. E2 contains 2 hydroxy group inside in the structure. So E3 contains the 3 hydroxy group. So it's a more hydroxylated, isn't it? Okay. Okay. So the one testosterone. Okay, I would like to highlight about the testosterone in terms of biochemical efficiency. Testosterone, dihydroxytestosterone is more potent than the testosterone. So action of dihydroxytestosterone is conversion is catalyzed by the 5-alpha reductase. Please be careful there. So you can see the hypothalamo, pituitary, gonadotrophic influence the production of sex hormone biosynthesis here in both male and female. Okay. Okay, so male testosterone level is about 9 to 30 nanomole per liter. And for the ladies, of course, the testosterone secretion is testosterone concentration in the female is very minimal. Okay, so it depends on the age. So from the teenage, and then the reproductive age gradually high, and then the andropause in male and menopause in female, the sex hormone secretion is declined. Okay, so the synthesis is similar. Okay, similar meaning to say that side chain cleavage enzyme will degrade the cholesterol to the pregnenolone, later progesterone, later DHEA, and later, and then they will form the androstenediol. And finally, 5-alpha reductase will catalyze on the testosterone and convert the most active, biochemically active form of testosterone is DHT and male target cell. Okay, females as well. So estrone, E2, E3, E1, right? So E1 is least abundant of the 3. And E2 is a primary estrogen of the ovarian origin. So present almost always reproductive female. Okay, E3 is abundant primarily in the placenta during pregnancy. This is how the what you call that, their concentration in our ladies will be different in terms of what you call that time. Okay, so estrogen are formed by the aromatization of androgen. That aromatization occur require the NADPH and oxygen. Okay. Okay, so postmenopausal women, conversion of estradiol to estrogen is E1 is the major source. The statements I've already mentioned recently. So E1 is relevant to the health and disease state because its conversion to the estrogen sulfate is a long life derivatives. Okay, so aromatase inhibitors here, as I mentioned earlier, can be treated in the what you call that gynecomastia, and also what you call that in the estrogen receptor positive breast cancer cases. Okay, so aromatase deficiency can lead to the virilization in the female. Okay, okay, so can be this overexpression can be characterized disease as cirrhosis of liver, hyperthyroid, aging, and obesity. You can find the overexpression of aromatase enzyme. So these conditions are related with the overexpression of aromatase. Okay, that's all about the adrenal cortical hormones and sex hormone. Last but not the least, so let me finalize the hormones of the adrenal medullars. Actually, this what you call the explanation, I think you have already received this the biosynthesis of adrenal medulla in year 1 biochemistry lecture, especially in the specialized product formation. So adrenal medulla, the starting material is totally different from steroid hormone. The starting material is tyrosine amino acid. So tyrosine amino acid transform the catecholamines in the chromaffin cells of the adrenal medulla. Okay, these hormones are very important for acute and chronic stress. You have already known that. But during stress response, they work together with the glucocorticoids, growth hormone, vasopressin, angiotensin 2, and glucagon together. So chromaffin cells synthesize the dopamine, but dopamine is mainly synthesized in the brain. But epinephrine and norepinephrine, especially among them, epinephrine is the major product. 80% of total hormone synthesized in adrenal medullas are epinephrine. Okay, so synthesis. You have already known that, so I will just revise brief. Okay, tyrosine here. Tyrosine is actually the non-essential amino acid that contain the hydroxy group in the side chain ring structure. So actually derived from the essential amino acid phenylalanine. So by the enzyme phenylalanine hydroxylase. So again, once tyrosine is formed, tyrosine undergo the hydroxylation reaction and the ring structure of the tyrosine, and then by the enzyme tyrosine hydroxylase form dopa. Once dopa is formed, the carboxy group may be released and removed, and then the decarboxylase enzyme activity. Decarboxylase enzyme remove the carbon dioxide. And then next step is form dopamine is formed. Dopamine beta hydroxylase also remove the addition of hydroxy group here and form noradrenaline. And methyltransferase enzyme transfer the methyl group and make the adrenaline finally. So that reaction, dopamine is also formed in the corpus striatum of the brain. So if something happened in the deficient synthesis of dopamine, may result in deficiency of dopamine and lead to the Parkinson's disease. Okay, that melanin can be formed from the dopa. Okay, so here, so biomedical importance of the tetrahydrobiopterin is very important for the conversion of phenylalanine to the tyrosine. So it's involved in the 2-hydroxylase enzyme, the tetrahydrobiopterin. Presence of tetrahydrobiopterin activate the phenyl hydroxylase and tyrosine hydroxylase. So that reaction also require the NADH, vitamin B3, very important. So you can see that the dopa is formed. You can see the dopa decarboxylase remove the carboxy group from the dopa, make it a dopamine. Okay, make it a dopamine. So next step is also hydroxylase enzyme. So we form norepinephrine hydroxylation reaction. That hydroxylation reaction require vitamin C. So that's why, especially, you know, that during the what to call the stressful condition, since we are encountering or facing with a stressful condition, or we are having fever, we are having the infection. So our dopamine and epinephrine or epinephrine requirement will be higher. So in that case, we also require these additional vitamin C supplement. Okay, so that's why you can refer the current situation. So sometimes we are advised to have a vitamin C supplement, right? Okay, to fight back the viral infection, COVID-19, so on and so forth. So detailed reaction explained here. Okay, so ring hydroxylation happens. So I would like to highlight that here, rate limiting enzyme. This is a rate limiting enzyme for the whole catecholamine biosynthesis. This enzyme activity can be inhibited by tyrosine derivatives methyltyrosine. So actually methyltyrosine is a drug, prodrug, can be used in the treatment of pheochromocytoma. Pheochromocytoma is a tumor, the malignant tumor of adrenal medulla. So in order to inhibit the overproduction of adrenaline and norepinephrine, so tyrosine derivatives methyltyrosine can be used to block the synthesis so that norepinephrine and norepinephrines are formed. It's a kind of treatment in a pheochromocytoma cases. Chelating enzyme, iron also can inhibit the enzyme activity here. And next step, a decarboxylation reaction of the dopa, make a dopamine, right? So in this state, I would like to highlight about that can be inhibited by alpha-methyl dopa and other related compound are 3-hydroxythiamine, alpha-methyl, tyrosine, and metaraminol. And these are the related compound alpha-methyl dopa. It also can be used in the treatment of hypertension, especially in the alpha-methyl dopa is used in the pregnant hypertension. Okay, pregnant lady with the toxemia of the pregnancy. Okay, halogenated compound also can inhibit the decarboxylase enzyme activity. So it's okay. Of course, the dopamine deficiency may lead to the Parkinson's disease. Okay, side chain hydroxylation reaction. This enzyme activity, copper is a cofactor for this enzyme. Fumarate is a Krebs citric acid cycle intermediate, is a modulator for the this side chain hydroxylation reaction. N-methylation is the last step. Formation of epinephrine from the norepinephrine. So it's ethanolamine and methyltransferase enzyme activity is very important. This enzyme activity is enhanced by the glucocorticoids. So that's why role of glucocorticoids in the biosynthesis of adrenal medulla is important, bearing your mind. So they work together. Okay, metabolism of catecholamine. So dopamine can transform to the norepinephrine. Catechol-O-methyltransferase catalyze the norepinephrine to chain normetanephrine. And monoamine oxidase catalyze the norepinephrine to form DOMA. In the biosynthesis, epinephrine is formed by the action of phenylethanolamine and methyltransferase activities. You can see that. In a catabolism, monoamine oxidase can be degraded, can degrade the epinephrine to form DOMA. Okay, catechol-O-methide transferase UMT also catalyze it and form metanephrine here. No metanephrine, metanephrine again. The monoamine oxidase and COMT catalyze and finally form the vinyl mandelic acid. So vinyl mandelic acid is the end product of the catechol-amine and they are excreted in the urine. So that the catechol-amine cannot cross the blood-brain barrier, hence especially dopamine must be synthesized locally. So this is a problem. So Parkinson's disease, which is you can see that Parkinson's disease is a long-term motor disorders, and then what you call that due to the local deficiency or dopamine synthesis. So in that case, levodopa, L-dopa is the precursor of dopamine, but it can cross the blood-brain barrier. So it's an important agent for the treatment of the Parkinson's disease. Okay, my dear sisters and brothers, that's all about today's lecture. Any questions so far? Any questions? Any questions? Any questions? Any questions?
No question, Doctor.
Any questions? Any questions, Sister Aisha? Brother Shamsuri? Sister Nur Aisha? Nur Atifa? Go in, long time no see. Is it going?
Yes, Linda Messi.
Need time to digest. Yes, actually, yes, Tawfiq. Yes, actually, very simple, you know, the starting compound is cholesterol. Cholesterol later changed to pregnenolone. So that's why, you know, even though there are many steps involved, but simple things, you know, simple meaning to say that I mean, the corticosteroids hormones are synthesized from the cholesterol. The cholesterol is important in the synthesis of steroid hormone. Since, you know, the many enzymes involved, the most of the enzyme are hydroxylase enzyme. So hydroxylase enzyme make sure the attachment of the hydroxy group in the steroid nucleus make it mineralocorticoids, glucocorticoids, and then the androgens, later sex steroids. Okay, so hydroxylation reaction require the what to call that require the NADPH as usual for the reductive biosynthesis. Yes, sister, brother Nabil, any questions? Come on. Any questions, brother? Congenital adrenal hyperplasia, yes. Yes, brother Nabil. Can you microphone? Yes, of course. Okay, sure, Nabil. Can you see the picture here? Okay, so actually, these are the biosynthesis of steroid hormones. So as I mentioned earlier, we require the many enzymes in each and every steps. First step is a 3-beta hydroxysteroid dehydrogenase enzymes activity. So if enzyme is deficient, could you imagine that? The what you call that the whole process cannot be proceeded anymore. So in that case, enzyme is blocked since at the beginning, early of the biosynthesis. What happened is that, you know, no production of glucocorticoids, mineralocorticoids, and also androgens or estrogens. I mean, the adrenal origins. So in that case, what happened is that, you know, male do not have enough androgen. So that's why, you know, the sex glands formation are not complete and they will present with a female-like genitalia. Okay. Okay. So very rare, as I mentioned earlier. Okay. And next one is this step. 17 alpha hydroxylase action. So you can refer to the biosynthesis slide. So progesterone supposed to change to the 17 alpha hydroxy progesterone, right? So if enzyme is deficient here, deficient in that step, okay, there are no longer complete the pathway here, consequent reactions, but the progesterone can escape to the this way, isn't it? So if 17 alpha hydroxylase defect is here, but 21 hydroxylase are maintain normal. So in that case, no sex hormone and cortisol production, of course. But here, 21 alpha hydroxylase is preserved normal. So that's why all progesterone, which are formed recently, the progesterone will goes to the what you call that alpha deoxycorticosterone formation, because your 21 alpha hydroxylase enzyme is normal. It's synthesized normal amount. So that's why as a result, will be increased production of mineralocorticoids. So as a result, of course, increase the sodium and water retention, and also increase the blood pressure. Of course, female-like genitalia were present in the male because there is no production of testosterone, since 17 alpha hydroxylase is blocked totally here. No production. Okay, but the major most common form of the congenital adrenal hyperplasias are related with the 21 alpha hydroxylase enzyme deficiency. So if 21 hydroxylase enzyme is totally deficient or lack, it will defect not only to the mineralocorticoids pathway, also affect to the cortisol synthesis as well. You can see that here, red color, block here, block here. But luckily, you can see that your androgen formations are what you call that successfully going on, isn't it? Because they do not rely on any 21 alpha hydroxylase enzyme activities, right? You can see that here. So that means that mineralocorticoids, glucocorticoids are virtually absent, isn't it? So overproduction of androgen leading to masculinization and external genitalia in female, and then early virilization in males. That means that those who are having this problem, the ladies, the female will have early menarche, may also have their what to call that adolescent. They reach adolescent earlier than the normal age. Means that some of the male and female, they start their what to call that reproductive age since maybe 7 years old, 8 years old, 9 years old, 10 years old, so on and so forth, something like that. Okay, so another enzyme defect is found in the alpha-1 beta hydroxylase deficiency. So that enzyme only affect to the cortisol and your mineralocorticoids. It doesn't have any action in the androgen production, right? So very clear here. So decrease the formation serum cortisol and corticosterone, so on and so forth, but increase production of here 11-deoxycortisol if in case 21 hydroxylase is preserved normal, that is increase the 11-deoxycortisol level. Okay, so that is what you call that, because this hormone suppress the renin angiotensin system, it causes the low renin hypertension. Okay, this is special. Okay, another one is that overproduction of this androgen will also happen because there is no interference in enzyme activity in the androgen synthesis. Okay, Brother Nabi, have you got it? Brother Nabi? Okay, thank you for the questions, Brother Nabi. Sorry, my microphone is broken, no problem. Okay, now finally you got it, right? So major problem that affect to the mineralocorticoids and glucocorticoid synthesis are due to the 21 alpha hydroxylase enzyme activities. deficiency. Okay, you got it. Yes. Okay, thank you for the questions and thank you for your interest. Yeah. Yeah. Okay. Go in. Any questions? Brother Shamsuri, any questions? Brother Arif, any questions?
So far, no.
So far, no, eh? Okay. Okay. So hopefully, you will, what you call that, you can answer the questions well in your quiz one. Okay. Okay. So any questions from sister? Sister Christina? Sister Shamimi? Sister Faiza? Brother Akmal? Okay, do not forget to sign in, sign the attendance. Because since we have to report the attendance report to the office soon. Okay. Okay. So if there is no question so far, let me stop here. Okay. Okay, thank you very much for listening.
Thank you very much, Doctor.
You're welcome, sister.
Thank you,
Atika.
Doctor.
Okay, you're most welcome.
Thank you, Doctor. Bye-bye.
Thank you, Doctor.
You are welcome.
Thank you, Doctor.
Thank you, Doctor.
If you have any questions, feel free to reach me anytime. Do not reluctant to call me. Do not reluctant to reach me via many means, right? Nowadays, Telegram, and then the what you call that, WhatsApp, email. Okay. Okay. Bye bye.
Bye-bye, Doctor.
All right, Doctor.
Take care, everybody.
Take care, Doctor.
Thank you.
Yeah.